Okay. So, do you ladies have any questions about some of these experiments that were done? Because I did not cover that. I went right over it. We talked specifically about replication, but there were some questions about experiments. Do you have any questions about that?
Sir, can you talk about number 2?
Yes. Okay. So that's I don't know if that's actually in your book. So this actually is very similar to a question that I gave you, which was about, if you recall, when we did feedback inhibition. Remember we looked at when an enzyme, sorry, let me just clear this. Hold on, what was that? Let me clear the canvas. We know we talked about like when an enzyme converts a product, like from A into B, into C, into D, and each step is controlled by an enzyme, right? Let's call that enzyme 1, call that enzyme 2, and call that enzyme 3, right? So in this step, let's say we take a product like A, a reactant, and we convert it to a product like D, okay? Each step is controlled by a specific enzyme. Now, do you remember we talked about what happens if there's like a mutation? Let's say imagine that enzyme 2 doesn't work, right? And the question, I think I even gave you a test question, was how do you figure out that enzyme 2 is the problem, okay? So in this case, what you would do is you would add substrate A, right? And you realize you don't get D because the problem is enzyme 2. Then you add substrate B, you still don't get D because B can't be converted to C because enzyme 2 doesn't work. And then you add substrate C, and then when you add substrate C, you get D because what you do is you overstep, go around enzyme 2. And what that does is it allows you to identify that this enzyme doesn't work, right? There's a malfunction. So the idea that, so the question was about beetle and tatum. They did experiments with certain fungi that basically led them to the conclusion that if there is a mutation in a gene, because look, these are all enzymes, and enzymes are, you know, for the most part made of protein, and we know that the instructions to make a protein come from the DNA, right? So if there's a mutation in that DNA, let's say that mutation makes the enzyme malfunction, so it doesn't work. Then what happens is that gene, that mutation in that gene causes the enzyme to malfunction, and you can pinpoint where that mistake is, right? Like what we talked about here with this feedback loop, right? The feedback being the D goes back and turns off A. So what Beadle and Tatum discovered was that mistaking one gene caused one enzyme in the pathway to not work. So that led them to the idea that one gene gives you the information to make one enzyme. Okay, that's the idea. That help?
Yes, sir. Sorry.
That's okay. That's okay. We are recording this, so that way, if anyone needs to go back to the other presentation, it's fine. Okay. Other questions from here?
Sir, can you also answer number 9?
Okay, so the question is, why is the code, why must it be made of 3 letters? Okay, that's an excellent question. It's actually going to be related to something we're going to talk about today. So let me pull up the genetic code for you. So the genetic code, let's just Google it. So genetic code. Okay. So here is an example of a genetic code. Okay, so let's take a look at, I think this one's from genome.gov. Okay, so here it is on the right. Okay, so one thing that you notice is the code, which is over here. So if I make it, let's see if I make it a little bit bigger so you can see it a bit better. Okay. The code is written in, it's called a codon. So codon is 3 letters, okay? Now, for example, if I look up here, you'll notice that the letter UUU codes for an amino acid. UCU codes for a different amino acid. CAA codes for a different amino acid, okay? Now, why 3 letters? It's because, think of the math behind this. Let me clear this. If I have 20, so how many amino acids are there? There's 20, right? There are 20 amino acids, okay, that living things, it's actually a little bit more than 20, but for the most, it's 20 amino acids that living things use to make protein, okay? Now, the codon is written in 3 letters at a time. So like UU would be, for example, a codon. It can't be 1 letter because there's only 4 letters, right? There's U, then there's RNA, right? U, G, C, A. So if a codon, which is basically the instructions that give you an amino acid, if it was written in 1 letter code, then the maximum number of amino acids you could get here would be 4 amino acids, right? Because U would give you 1, G would give you another 1, C would give you another 1, and an A would give you another 1. But there's 20. So the genetic code cannot be written with 1 letter. So 2 letters actually wouldn't work either. Like, for example, UU cannot be a code, UG cannot be a code, UC, UA, because if it's made of only 2 letters, then the math says that there is 16 possibilities, okay? Because there are 4 codons and there's 2 in this example. So 4 to the power 2 is 16 possibilities, but that is still less than the 20 amino acids that we use to build protein. So it was thought that it's probably written in 3s, because if we write the letter or the codons in letters in groups of 3, like for example UUA or UGA or UCG, for instance, or UAG, well, mathematically that would give you 64 possibilities, which is more than enough to code for your 20 amino acids. So the letters have to be written in 3s, because that would give you enough combinations to give you the instructions to make the 20 amino acids. Okay? Did I clarify it?
Yes, sir. Yeah. Thank
Okay.
you, sir.
No problem. Any other questions from this unit so far? I mean, we can always go after, we can always come back to this. Okay, so then if no more questions, what I'm going to do is what I did was I took some of the questions from Schoology. There were a few interactives where you had to watch some videos and you had to answer these questions. So one of the questions we'll look at is the role of complementary base pairing in genetics. These are questions specifically in relation, well, the first 4 is in relation to replication, okay? So I wanna just quickly review that with a couple of really short video clips. So here's the first one. I'll narrate it for, oops. Oh, sorry, it's not working. Sorry. You girls can't hear the audio of the video when it's playing, right? Let's see if this works. Oh, that's not working either. Trying to find the right media player. Let's try. None of these are working. Hey, what's going on here? None of these are working. I'll have to play it then in the actual Schoology folder. So let's try that. Hopefully this, it's been pretty good. So let's try this. Okay, so here's the first one. So this is replication. So you can see here's your double helix, right? And we know what has to happen. We know that the first thing that has to happen is we need to open it up, right? Because to be able to copy the DNA, it has to be opened up so we can expose the template strands, right? We talked about this last time. And we know when it gets copied, what happens is we use a complementary base pairing, right? And we start with primers, if you recall. So we use, for example, in this case, if we read the letter C, we put in the letter G. Okay, that's what we talked about last time. Okay. Here's just a follow-up video clip. So in this clip here, we've already added the primer, right? The primer, remember, is the part that has to be put down first so that the RNA polymerase can build off the exposed 3' end using complementary base pairing. Okay, so this is a review from last time, and you can see you just add the complement, right? Where you see the letter T, you put in an A, you see the letter C, you put in the G, and you'll notice that the end is 3' because we're building 5 to 3. Okay, that's what we talked about last time. Okay. That's called the daughter strand. That's the strand that you're making from the template strand or the parent strand. Okay. And then the last clip is this one. So here you're seeing it, the direction. So we talked about the first thing that has to happen is you have to open it up. Now remember we talked about this gives you something called a replication bubble. This is your fork on either side, and the fork opens up in both directions. You can see that. So here comes one strand, and you notice the other strand is being built in the opposite direction. So this one is being built 5 to 3. That one's being built 5 to 3 because that's how you build. You build 5 to 3, you read 3 to 5. Let's keep going. So you'll notice this strand is just following, because this fork is opening this way. This strand just follows. This strand is just following this fork. These are called the leading strands, right? It's being built 5 to 3. Now, the other strand, that's going to be your lagging strand. So the lagging strand here, and there's going to be a lagging strand over here. And you'll see that in a second. There you go. There's the primer. And there's the other primer. You notice that it's going this way, away from the fork. And this one's going this way, away from the fork. There's your primer. There's some more primer. And then we start putting in some DNA. 5 to 3. right? So this was the leading strand because it's going with the fork. This is the leading strand going with the fork. The lagging strand is going away from the fork. So this one and then this one. You just keep building it. Okay. So you notice the leading strand just follows, but now the lagging strand has missing sections, which we need to go back and fill in. Which you saw before in the previous animation, again, is all using complementary base pairing. Okay, that was replication. That's what we talked about last time. So today what we're going to do is we're going to compare it to transcription. Okay, any questions before we move on? Okay, so let's look at transcription then. Because what happens in transcription is you're taking the DNA, which is found in the nucleus of eukaryotic cells, and you're going to convert that DNA into RNA by looking at specifically copying a section of the DNA called a gene, okay? And then the RNA transcript is going to be brought out of the nucleus and be used to make some sort of a protein, okay? That's the big idea. This is actually known as the central dogma of genetics, where you go from DNA to RNA, and then RNA into a protein. The going from DNA to RNA, that's called transcription. So replication was DNA to DNA through the use of proteins like DNA polymerase. Transcription is going from DNA to RNA, okay? And then RNA to protein, that's translation. So the central dogma of genetics is one of your questions in your course outline, is the way information flows from DNA through transcription into RNA, and then through translation into protein. That's called the central dogma of information flow. What we're going to do is we are going to compare that to, we're going to compare right now transcription to the way it works in replication, okay? So here is the video clip on specifically transcription. So in transcription, one difference right away is you don't copy all the DNA. You're only going to copy the section that is a gene, okay? So the first question is, where do you find this gene? You're doing a lab right now on the virus COVID-19, and you'll see that there are specific genes and specific sections in the virus, okay? Now, the viral genome mostly codes for protein. The human genome, only about 2% of it codes for protein. So these genes are scattered throughout the genome and they have to be found. So the way they're found is there's a stretch in front of the gene. It's called a promoter region, okay? It's a stretch of nucleotides, of sequences that instruct a specific enzyme, which is this one here in pink, called RNA polymerase, okay? So RNA polymerase is going to look for a stretch of DNA in the promoter region, and it's going to use that as a way to identify where to start copying the genes. Now it needs help, okay? So the transcription factors are other proteins that actually help RNA polymerase bind to DNA so that it can start its job of taking the DNA and transcribing it into RNA. And you're seeing that here. Now, one difference between transcription and translation, there's actually a few. One difference is that RNA polymerase can actually open up the DNA sequence by itself. If you remember in replication, there's another enzyme called the helicase that actually opens up the DNA strand. Okay, RNA polymerase can do it on its own. Sometimes it needs a bit of help, but it can pretty much do it on its own. The other difference is in replication, you want to copy all the DNA, so you're going to copy both strands. But in transcription, you're only copying one of the two strands where that gene is. So wherever that gene is, it's not going to be on both strands, only going to be on one strand, right? And you're only going to copy one of those two sections. And you can see that here in this animation. You'll notice nothing is happening down here because this transcriptional unit is up here. Now, in terms of similarity, the directions are the same. So you read 3 to 5 and you build 5 to 3. But you'll notice that there is no color difference in this animation because there's no primer required, okay? RNA polymerase not only can open up the DNA on its own, but it also does not require a primer. It does not need to see that 3' end. It can start from scratch, okay? So there are ways to compare replication to transcription. Questions? Next clip then. So we just left off saying that the directions are the same. The role of complementary base pairing is exactly the same as you saw in replication, okay? The way you build RNA from DNA is through complementary base pairing, just like the way you build a daughter strand from the parent or template strand. You read a letter, you put in the complement. So you read the letter A, you put in U. You read the letter C, you put in G. Remember, there is no T in RNA. But again, if you go back here, you'll notice these are all complements, right? So if you read the letter, for example, this is a T, you put it in an A, but if you read an A, you put in the U. Now, eventually, the gene is done. So that's when transcription stops. So again, because it's not replication, you're not copying everything. You're just copying a section, which is that gene. This thing that you copied is called mRNA. It's actually called a pre-mRNA because what you'll see is it's actually going to get modified a little bit. Now, what's interesting is that this thing that we just made here, this is actually, when you do your COVID-19 lab and you look at the coronavirus, the genome of the virus is an RNA genome. And the actual genome itself, because it's an RNA genome, actually looks like this mRNA. It's very, very similar to it. And the role of the mRNA is to instruct the building of proteins. It's actually one of the questions you'll have to look at in your lab is looking at the genome of the virus. It's actually the same as mRNA. The only difference is that unlike what happens in eukaryotes, the mRNA in COVID-19 really doesn't go through too much change. You'll see here that the mRNA in eukaryotes actually gets modified. So there's a couple of things that happen. So one thing you start to notice is that something happened at the beginning of the message and something's happening at the end of the message, okay? If you look at your RNA genome for COVID-19, you will see a long stretch of A's at the end of the viral code, because that is actually just like the poly A tail that you're seeing here. There's also here, we see something called a cap. So the cap and the tail, the function of these two things is actually protection. If you remember, this is made in the nucleus and then it has to leave the nucleus. The nucleus, outside the nucleus is actually like it's kind of a hostile environment. So the cap and the tail serve as protection. So what's interesting is if you look at the COVID-19 virus, there is a poly A tail, and I can't remember if there's a cap. There might be a cap, which kind of makes sense because the viral genome has to be protected from from the hostile environment of the cytoplasm. Otherwise, it will get broken down. So you will see a poly A tail towards the end of it. I can't remember if there's a cap. You have to double check on that one. But the following thing does not happen in the viral genome, which is something called splicing, which you're going to see here. So the viral genome does not contain regions that get cut out. And those regions that get cut out, they're called introns. And you're going to see that in the next clip, which is, I think is this one. It should be this one. Okay, so the RNA that's made is not fully mature. It's called pre-mRNA. It gets modified a little bit at the ends. The 5' and the 3' end get modified. And that's for protection. Then what happens in eukaryotes is that there are regions in the mRNA called introns that actually get, you're going to see here, they get cut out. And then what happens is the exons get put back together. And you can see that here in this clip. Once that happens, now the mRNA is fully mature. Now, the viral genome does not have this. Okay. The prokaryotic genomes, bacteria don't have this either. So the question is, what is the advantage of having this? Let me show you something that is quite interesting. I'm going to show you a gene. The name of the gene is kind of cool. It's called ABCD1. And the gene is quite large. So here's the gene itself, okay? You'll notice some of the terminology, 5' UTR, and you see that terminology when you look at the viral genome, okay? So what you're seeing here is the genome for the gene for a protein called ABCD1, which is it codes for a transporter. Okay, it's a protein that allows for the movement of things through a membrane. Okay, now the size of this gene is quite large. It's 19,900 letters practically, right? Now, the COVID-19 virus is about 30,000. So this gene, this one human gene, is about 2/3 the size of the viral genome in COVID-19, okay? Now, you see these regions called IVS? Okay, these regions, IVS over here, these are the introns, okay? These regions right here, I don't know exactly where it starts and where it ends, but you notice that they sit in between the exons, right? So those regions get cut out. Some of them are quite large. You can see here's another region. Here's IVS. So this is intron 4 and sits between exon 4 and exon 5, and it gets cut out. Here's intron 3, and it sits between exon 3 and exon 4, and it gets cut out. Now, why are we doing this? Why are we cutting out introns? So here's the idea, okay? Imagine the mRNA is like this. I have exon, I have an intron, I have an exon, I have an intron, I have an exon, like that, okay? Now, imagine I call this exon A, B, and C, okay? Now, what happens in prokaryotes is there are no introns, and in viruses, there are no introns, so what I'm going to show you does not occur, right? What I'm going to show you is called splicing, because it gets cut, okay? So in eukaryotes, the introns get cut out, and then what you do is you put together the exons, like this, okay? Now, that's not the only way you can do this, because otherwise there would be no point in just copying. I mean, really, what's the point of copying all those letters? I mean, if you go back to this and we look at, hold on a second. if we go back to this and we look like, what's the point of copying all these letters in the intron and then getting rid of them? Seems like a waste of time. Here's the advantage. You don't have to put the sequences like this. Because you're doing some splicing, I call it like, I think of it as like cutting and pasting, okay? You don't need to cut and paste in the same order. You could do this. You could do EA, EB, EC. You could do EC, EB, EA. I guess you could do EC, EA, EB. Now, what's going on here? What's going on is because we're doing some cutting and pasting, each of these gives you a new protein. So we'll call this protein 1. This will be protein 2. This will be protein 3. And this will be protein 4. So the advantage of cutting and pasting, even though you're copying introns and then discarding them, right? The advantage of that is what you can do is you can create more proteins really from the same gene by doing this cutting and pasting. Now, so that seems like a huge advantage, but remember, it comes at a cost, and the cost is that you need to copy things called introns, which, oh, I think I just realized how you can read this. The introns are actually in lowercase letters. Look at that. Yeah, the introns are in lowercase letters. We can actually identify where they start and where they stop. Just notice that right now. So I guess the cost is that you're going to have to copy letters that you're going to eventually discard. And the things like prokaryotes and things like viruses, they want to have, not they want to have, but they have a very, very small genome. So for them, this would be, I guess it would just be a bit too difficult to manage, because this would require a larger genome because you have to copy more information before you can cut and paste. Let me show you like an example of kind of like an analogy, I guess, what the advantage of something like this would be. Because I can tell you that it gives you more protein, but let me show you something really cool that it's not my own. I didn't come up with this, but getting more proteins from the same genes would be analogous to reading something like a message, but getting a different message from it. So let me show you what I mean by that, okay? So let's say I gave you something like this. This is not mine, so I'm not, I didn't come up with this. But let's say I wrote this. And this is not mine. Okay. So prokaryotes and viruses would simply read this only one way because they don't cut and paste. Okay, so they would read it in, and I don't know how you're going to read it. Oop, where'd it go? What just happened there? It just disappeared. Wow. Okay, let me write that again. I think I actually forgot to write something. Sorry, I forgot to write the letter A. Okay, so because viruses and prokaryotes don't do any splicing, they only read their genome one way, okay? And the advantage of that is you save a lot of space because you don't have all those introns you copy. The disadvantage is that because you're only reading the genome one way, you cannot create new messages from the same gene like you can with splicing. So what eukaryotes can do is they can take a message like this and they can read it different ways. So I want to volunteer. Can someone read what this message says? Anybody? Chelsea, read it.
A woman without her man is nothing.
Chelsea, I can't hear you.
No, I don't want to read it.
I can't hear anybody. What's going on? Oh, Chelsea. So, you repeat that again?
A woman without her man is
Hi,
nothing.
sister.
Say it one more time. I just realized my mic was all the way down. My volume was all the way down. So were you guys asking me questions before?
No, we could hear you.
No, but were any of you asking me questions and I didn't respond to them?
No,
No. Okay.
we're all
Okay.
muted, so
Okay. Okay. I turned the volume down because what happens when I play video, you can't hear the video. So I just turned the volume all the way down. But I forgot to turn it back up. So when Chelsea answered, I couldn't hear her. So Chelsea, one more time, read that message.
A woman without her man is nothing.
Okay, that's one way to read it, right? Can I show you a different way to read it? Okay, I'm going to do something, okay? Now I'm going to add some commas. Okay, I added some commas. Now, can you read it again?
Oh my God, that's not.
Read it now.
A woman without her man is nothing.
Now, isn't that the opposite meaning? So before it was a woman without her man is nothing, and now a woman without her man is nothing. That's a totally different message, right? And this is not mine. I saw this from a TED talk by a guy named Juan Enriquez, and I said this is brilliant. I'll show you another one, okay? Here's another one.
let's eat grandpa.
Yes, you remember that one, right? Okay, read that. Renee, read it.
Let's go together.
Yeah, of course. Let's go together, right? Yeah, because you're
Let's go
nice.
together.
You're a nice girl. You would say, "Let's go together," right? But what if I did this?
Let's go to get her.
Yeah, let's go to get her. That doesn't sound so nice, does it? Let's go to get her. That doesn't sound as nice as let's go together, right? And what was the example you gave? Let's eat grandma. Let's eat. No, wait, is it? Yeah. So let's eat grandma, right? If I put that comma in, let's eat grandma, as opposed to let's eat grandma, like the big bad wolf, right? So, okay, so that's the advantage of splicing. Is splicing allows the cell to rearrange the exons so that you get more than one message from the mRNA. Okay. Okay. So let's go back then to transcription. All right. So this is the end of the video. Okay. So with regards to transcription versus replication, okay, let's look at this section here, comparing transcription and translation. All right. So sorry, that's supposed to be comparing transcription and replication. That's a mistake. That's supposed to be transcription and replication, okay? Comparing the two. What was the name of the enzyme that does the copying in replication? So what enzyme copies in replication and converts DNA into DNA?
DNA polymerase.
Very good, DNA polymerase, excellent. What is the name of the enzyme that copies DNA into RNA? It's not DNA polymerase, it is?
RNA polymerase.
Yeah, RNA polymerase, excellent, okay. What about primers? Does replication require primers?
Yes.
It does, because DNA polymerase does not know how to start. But as you saw in the video, transcription, RNA polymerase does not require primers. It actually knows how to start from scratch, okay? In terms of reading and writing, do they both read and write the same way? If you remember DNA replication, it's read 3 to 5 and built 5 to 3. What about transcription? It's exactly the, yeah, it's exactly the same. Okay, the reading and writing are exactly the same. In terms of the number of strands copied, okay, in replication, we copy both strands or just one strand?
Both.
Yeah, because you want to copy everything, right? You want to replicate all the DNA. But in transcription, we only copy what?
One.
Yeah, because the gene is going to be on only one of the two strands. What is the role of the promoter region? What's the role of the promoter region? So imagine that this is your DNA strand. Okay, we know that DNA is double-stranded, right? So the gene is only going to be, let's say the gene is here. Okay, call that gene A. Maybe there might be another gene over here. We'll call that gene B. Maybe there's another gene over here. Call that gene C, right? Let's say that this is the 3' end and this is the 5' end. Obviously, then this would be 5' and this would be 3', right? How does RNA polymerase find gene? Like, how does it find it? It has to find something before. And that something before is called the what?
Primer.
Not primer. It does start with the letter P. It's called the promoter region.
Promoters.
Right? So what it does is it scans the DNA. Because remember, the read, it's reading 3' to 5'. So it starts scanning 3' to 5'. And when it finds the promoter, it knows that it has to copy the gene because it's going to be located right after the promoter, right? Here, it starts scanning, and then when it finds that promoter, it knows it has to copy that gene and convert it into RNA. Okay? All right, let's look at then translation. Okay? So translation is when we go from the RNA after it's mature, after it's been spliced up in eukaryotes, but not in prokaryotes and viruses because they don't have introns. And we go from the RNA into some sort of a protein, okay? So let's take a look at that right now. I'll put the volume down. Well, actually, I'll put the volume a little bit down. Okay, so here we go. And if you have any questions, jump in, okay? So here is just simply, your amino acid list. We talked about the fact that the amino acid list comes from, or it's encoded in your DNA. And if you recall from your biochemistry, this is an amino acid and that's an amino acid. And the only thing that makes them different are the R groups. And the way you attach them is one carboxyl end would be attached to the amino end through a bond called a, what's the name of that bond? Plus the P.
Peptide.
Yeah, peptide bond. Very good, okay? Called the peptide bond. So let's take a look at actually how this works. So let's take a look. So we know that this is like, you're gonna see transcription. So we take the DNA, we know we copy it into mRNA through complementary base pairing, right? Through the enzyme RNA polymerase. Then we take the mRNA and we're going to translate it, okay? Now you'll notice one thing right away. Some of the RNA code or some of these letters don't get converted into an amino acid. Okay. You notice how the amino acids line up with 3 letters? So like this letter AUG lines up with this amino acid, and then CAG lines up with that one, and then GUU lines up with this one. But you notice that nothing lines up with this. Okay. And that's because not all of the RNA actually codes for amino acids. So when you look at the COVID-19 viral genome, you notice that if you remember, the protein, where we start making protein, actually does not occur at the beginning of the genome. There's a stretch of about 200 and something letters that is not actually converted into any protein, okay? That's actually called your leader sequence or your 5' UTR. The UTR standing for untranslated region. So it's not translated into an amino acid. So let's take a look at how this process actually works. There's your codon. And remember, it has to be 3 letters because 1 letter is not enough, 2 is not enough, but 3 letters gives you 64 possibilities, which is more than enough for your 20 amino acids, right? Okay, so this is something called an amino acyl tRNA synthetase. This is something that actually brings in the amino acid. But what's really interesting is the way it does it. If you look at, so this is, let me actually explain this. So tRNA, sorry, not amino acid, tRNA synthetase. So let me explain that. Hold on, just got ahead of myself there. The let me take a screenshot of this. Yeah, it's probably better. Take a screenshot. Okay. Let me put this up. So what you're seeing here, this thing here, this is called tRNA. Okay. There's an enzyme called an amino acyl tRNA synthetase that actually attaches the amino acid to the tRNA. Okay. So this is the tRNA itself. Okay. Now the T stands for transfer, because what it's doing is it's transferring the message basically of mRNA into some sort of an amino acid sequence by bringing in the actual amino acid. And the way it does it is, if you look closely, the tRNA is basically a sequence of RNA, right? So it's made up of the letters A, U, C, and G. And there's a region called an anticodon, which you can see right here. And you'll notice, so here's my anticodon, it's right here. And you'll notice something about the anticodon and the codon. What do you notice? What do you notice is a connection between these two things? Anybody? What's the connection between the anticodon and the codon? Christine, what do you notice?
So I'm not sure, but are they base pairing or?
They are, yeah, very good. Yeah, you'll notice that there's complementary base pairing going on, because look what you have. You have A matching up with U, right?
Mm.
And then you have U and A match up again, and then G and C. So this is interesting because now you've seen complementary base pairing in DNA replication, right? Because remember, in DNA, the idea in replication is you read the letter A, and you use that to build what? T, 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 right? You saw it in transcription, So if I read the letters AAA from from DNA, and that would be transcribed into what? Into UUU. But you're also not seeing it in translation. So this process of complementary base pairing is used in replication, transcription. It's also used in translation, where the anticodon matches up with the codon. So complementary base pairing is a very, very important concept in genetics. Okay, so let's take a look at what happens. So there's your tRNA. So you can see the complementary base pairing being highlighted. Okay, so let's take a look at the next clip. So here's the tRNA. Okay, now it's what is it? It's just a twisted strand of RNA. Okay, twisted strand of U, A, C's, and G's. All right, that has a 3' end. Therefore, if this is 3', what would this end be over here?
5.
Yeah, yeah, it's no different than you saw with DNA, right? One end is 3', the other end is 5'. Now, this is an amino acid. So if we wanted to bind the amino acid to the protein, okay, so let's say this is my amino acid and that's my RNA molecule, my tRNA. Here's the trivia question. You should remember this from biochemistry, okay? How do I, or what do I need to remove to attach that amino acid to
Water.
very good water? Excellent, excellent, through a dehydration reaction. So that's where that enzyme, it's called amino acyl Trna Synthetase. You don't need to spell it. That enzyme does this reaction. It takes away water and allows them to bind together. There you go. Now it's bound. So these are your ribosomes, and this is actually where the translation is going to occur. It's going to occur in these organelles, which I think you studied these like way back and maybe even in elementary school. So how does this work? So this is the division of translation, the different parts, or different stages actually. So let's see how it starts. Again, notice that it's not going to use all the RNA information. There is a section called a leader sequence that does not get translated, okay? If I go back to my gene here from ABCD1, you'll notice that the exon starts somewhere here. So this section up here, this part, I don't know exactly where it ends, but that doesn't get translated. So that would be your leader sequence. That's the part that in this animation, the ribosomes do not convert into amino acids, okay? Aha, so AUG is actually very special. AUG is actually what's called the start codon. If I look at the genetic code, you will see that AUG, which is, let me make this a little bit bigger so you can see it. AUG, which is right here, is an amino acid that codes for something called MET, which is methionine. Okay, methionine is the first amino acid in translation. So it is the start of translation. That 3-letter code is a very special code. So you see the attached to complementary base pairing. Now remember, this is the tRNA with the amino acid attached to it. Now the ribosome has come together. Now, what you notice is in this ribosome, there are 2 empty slots. These are the sites where the tRNA goes into. You'll notice that this empty slot has a codon exposed, CAG. So, trivia question, what should the anticodon be of the tRNA molecule that is going to enter that section? Dorcas, what's the answer?
GUC.
See if you're right. GUC. There you go. You're correct. Here it comes. There's GUC. So what you're seeing, this is translation. This is where we go from the codon to an amino acid sequence. The tRNA is involved because it's using the anticodon to bring in the correct amino acid. Okay, you can see that here. Now, take a look at the way the amino acids are oriented. This is the amino end. This is the carboxyl end. This is the amino end. So what you're going to see is a bond form between the carboxyl and the amino end, right? By breaking this bond right here. So now that we have this amino acid is now attached to this one, and there's your peptide bond. Now watch what happens, and this is something called translocation. So now this tRNA is free because this amino acid has now been linked to this one. So this bond has been severed. So now it's going to leave to go pick up probably another amino acid. So watch what the ribosome does. It's going to shift. You're going to see it shift. Now this is called translocation, okay? Now by shifting, you'll notice it's exposed a new site. So now a new tRNA can come in with the codon CAA. I think it comes in now. Here it comes. There's CAA. Okay, so what then, if we bring this home, last clip. Okay, the last clip is this one. You can see then the same process is going to happen. This amino acid will be linked to this one. This bond will break. It'll be linked. So they'll be linked to this one. This tRNA is free, so it will leave. The ribosome will shift. Now, the next one will come in. I have a problem with this in the animation. Here's my sticking point. You'll notice the codon is GUU, and this one is GUA. What's interesting is if you look at the codons, so GUU is, where is it? GUU and GUA actually give you the exact same amino acid. So in the genetic code, there are often repeats where more than one code gives you the same amino acid. We'll talk about the significance of that when we talk about mutations. So because it's the same amino acid, and this is just a bit of a sticking point, if you really want it to be accurate, these two amino acids should have used the exact same color because they are the same amino acids. Now, that's okay. It's not a big deal. So what happens is this just keeps repeating, right? Next one comes in. You can see they get linked to the new amino acid. This bond breaks. It gets free, and it shifts over until it gets to a very special codon, which you could see in the genetic code. There's 3 of them. They're called stop codons. So there's one start called AUG, and there's 3 stop. Okay. UAA, UAG, UGA are all stop codons. And you can see that here, that this one that's coming up is UGA, which is actually a stop codon. So now what happens is another protein comes in and actually breaks up this complex, okay, adding water to break a molecule. We talked about that in biochem. So now we've released the polypeptide. I'll just skip ahead a little bit. One last little word is you don't need one ribosome to read the RNA. You could have multiple ribosomes read it because think of it like chefs, right? If you have more than one chef reading the recipe, think of the RNA as a recipe to make something. The more chefs you have, the more ribosomes you have reading it, the more protein you make, right? It's just a simple idea. Okay, so that is translation. Okay. So let's wrap this up really quickly and look at these few questions. And next time we come back, we'll look at a comparison. Okay. Talk about like you know, the connection between all these ideas. All right. So the last little thing we'll talk about is looking at transcriptional modification and translation. So in your own words, just whenever you think you have the answer, what is the role of tRNA and what is the role of mRNA? Anybody? Anyone want to try this? Adele, you there?
Yes, sir.
Hey, what do you think? So which one of those two is the actual code for the amino acid sequence? And which one is the one that brings in the amino acids? I'll give you a hint. The M stands for message.
Mm-hmm.
So which one do you think is the actual, like, well, giving you the answer, right? So what's the difference between these two? What does mRNA do?
It brings in the...
That's what this does. tRNA is what brings
Oh,
in
yeah.
acids, right? So in the video clip, the tRNA is what brings in the amino acid, but the mRNA is what determines the order of amino acids. That means the sequence, okay? If I look at the next question is, what is the role of the leader sequence? So does it get translated? Does the leader sequence get translated in genetics? What do you think? Yes or no? So the leader sequence is, I think it's in this clip. Hold on. It was in this clip. Is it now? Trying to find it where they actually put the word leader. I don't see it in it. The leader sequence would be, it's weird, but I saw it. So it's anything before AUG, okay? So it's before AUG. So what do you think the answer to that question is then? So does it get translated? The answer is no. So then what does it do? I would suspect the answer would be it's just simply something for the RNA to grab onto. It doesn't get translated into an amino acid sequence, but the RNA has to grab onto something so that it can hold the mRNA and read it. Okay, so it doesn't get translated. It's just like a handlebar that the ribosomes could use to start translation. For C, the role of capping, tailing, and splicing. Capping and tailing are for protection. And we talked about splicing is to rearrange the genome so you get more messages out of the same gene. The role of start and stop. Start is to begin translation. There's only one start codon, right? AUG. And stop is to stop translation, right? What are anticodons? These are these are found on your tRNA, and these are complementary to the codons. Okay, these are what allow the tRNA to properly bind to the codon so that they bring in the correct amino acid in the correct order. All right. And the last question is, what are ribosomes made from, and what is the ribosomes in translation? So ribosomes are made of two things. They're made of protein and they're made of RNA. Okay, so there's actually three types of RNA. There is mRNA, which is the message. Then there's tRNA, which brings in the amino acids, and then there's something called ribosomal RNA, which actually makes up the ribosomes. The ribosome is made up of rRNA, ribosomal RNA. It's also made of protein. Okay, so this thing is made up of two different biochemical molecules, RNA and protein, ribosomal RNA. And its role is to allow for the tRNA to correctly enter and bind to the codon so that it could properly translate the amino acid or the codons into the right amino acid sequence. Okay? All right, girls, how you doing? I know that was a lot. And I'm actually going to stop recording at this point. So, do you have any last minute questions before we end? That was long. It was like an hour and 20 minutes. And I'm tired. It's my third Zoom session today, by the way.
Third?
Mm-hmm. I'm gonna have one more in 10 minutes. Okay, so why don't we do this? Why don't we end here? Next week we will, as I said that when, as we go through this, you'll learn more about the lab I gave you. The lab I gave you, there's a lot in there. And there's a lot of stuff that's kind of complicated. The first part you can probably just do, but then it starts looking at some stuff where you have to analyze the genome. So it'll be a bit more, it'll make a bit more sense next week when we start actually looking at a gene, okay? And then we'll relate it to the virus, see how they compare. I'll send some information over the weekend about next meeting, next topics, and next steps. Other than that, have a great Good Friday. Have a great Easter. We'll talk later. Okay, girls?
Bye.
Meeting, sir.
Okay, ladies. Talk later. Bye.
Bye, sir. Bye.